Sampling theory. This is the topic of biostatistics and research methodology and applied mathematics, biostatistics. Content of this topic includes basic concept of inferential statistics, population, and sampling, sampling methods, and sample size. Inferential statistics. Synonym is inductive statistics. Inferential statistics is the logic of drawing statistics with valid conclusion about the totality of cases termed as population in any experiment, on the basis of examining a part of the population termed as sample. Inferential statistics draws broad conclusions for a population based on analysis of a smaller sample of data. Population. Synonym is universe. Definition. Group of individuals under study in statistical investigation is known as population. Population is a large group from which the sample is drawn. Classification based on number of items. If a population consists finite number of items, is known as finite population, for example, population of tablets of any batch. If a population consists infinite number of items, is known as infinite population, for example, population of weight of tablets. Classification based on object. If a population consists imaginary object, is known as hypothetical population, for example, population of throwing a die, the titration of sample. If a population consists concrete object, is known as existent population, for example, population of diabetic patient. Sampling. Definition. A finite set of the population selected from population with the objective of investigating its properties is called sample. Number of units in the sample is called sample size. Sampling is a tool which enables us to draw conclusions about characteristics of the population after studying only those items that are included in the samples. Factors to be considered when devising a sampling scheme include desired precision. The accuracy and precision desired will be a function of the sampling procedure and sample size. Best estimate or maximum information of the population parameters. The cost of sampling in terms of both time and money. The nature of the population, like diversity, lies between sampling of packed pharmaceutical formulation for testing and sampling of population with any disease. Sampling methods. Classification. Sampling methods are classifying into two major types: nonprobability sampling and probability sampling. Nonprobability sampling includes convenience sampling, purposive sampling, snowball sampling, quota sampling. Probability sampling includes random sampling, systematic sampling, stratified sampling, cluster sampling. Nonprobability sampling or authoritative sampling. Samples are chosen based on subjective judgment, rather than random selection. Convenience sampling. Samples are selected by an arbitrary method that is easy to carry out. Samples often refers as grab bag samples. Used when it is very difficult or impossible to draw a random sample. Results of studies based on convenience samples are descriptive and should not be used to draw inferences about the population under study. Examples are when sampling powder from 10 drums out of 100 drums. Those drums that are most easily accessible might be the ones chosen. When sampling tablets from a large container, we may conveniently choose from those at the top. Purposive sampling, also popular as judgment sampling, subjective sampling, expert sampling. Sample is chosen on purpose instead of randomly. The researcher relies on their discretion to choose variables for the sample population. Here, the entire sampling process depends on the researcher's judgment and knowledge of the context. Example is selection of patients in clinical trials. Only type 1 diabetes mellitus patients diagnosed within the last 6 months are selected for in clinical study. Snowball sampling. Snowball sampling technique works like chain referral. After observing the initial subject, the subject will identify people with a similar trait of interest. Researchers use this sampling method if the study-related sample is very rare or is limited to a very small subgroup of the population. For example, study on a rare disease. It will be difficult to obtain subjects. It is also possible that the patients with the same disease have a support group. Being able to observe one of the member as your initial subject will then lead you to more subjects for the study. Quota sampling. Quota sampling is a non-probability sampling method where sample is made by population characteristics like age, gender, by setting specific, predetermined quotas, mean strata. It involves selecting participants non-randomly to fill these quotas, offering a fast, cost-effective way to ensure subgroup representation. For example, pharmaceutical companies use quota sampling to collect feedback from specific patient segments, like diabetics, allergy sufferers, to evaluate new medication branding or packaging. Disadvantages. Non-probability sampling technique have a hidden bias. It is not possible to apply typical statistical methods to estimate the population parameters, like mean and standard deviation, and the precision of the estimates. This sampling method should not be used unless probability sampling methods are too difficult or too expensive to implement. Probability sampling methods. Probability sampling is bias-free sampling, sampling performed with specific procedures, which will ensure that every unit of the population consists of one fixed probability being included in the sample. Random sampling. A random sample is one in which each individual or object in the population to be sampled has an equal chance of being selected. Simple random sampling is most effective when the variability is relatively small and uniform over the population. For example, in quality control analysis, pick a small quantity of powder from top, middle, and bottom side of container. Stratified sampling. Stratified sampling is a procedure in which the population is divided into subsets or strata, and then random samples are selected from each strata. For example, the whole population is divided into homogeneous strata according to demographic factors such as gender, age, or diagnosis. Then the researchers draw a random sample from the different strata. Systematic sampling. Systematic sampling is a type of probability sampling method in which sample members from a larger population are selected according to a random starting point, but with a fixed periodic interval. It also results in a uniform sampling over the fixed periodic interval, which may result in a more precise estimate of the population parameters like mean and standard deviation. For example, in QA analysis, 10 samples are taken at a particular time every hour. During a production run, each time duration, sampling may be chosen in a random manner. Cluster sampling. The population is divided into groups, or clusters, each of which contains subunits. In single-stage cluster sampling, clusters are selected at random, and all elements of the clusters chosen are included in the sample. Two-stage cluster sampling may be used when there are many primary units, each of which can be subsampled. For example, a 10 lakh tablets batch, packed in boxes with 100 tablets, randomly selecting 100 boxes and then inspecting 10 tablets from each of this box. In stratified sampling, there is homogeneous subgroups, that is, strata, and sample made from a fraction from all strata, while in cluster sampling, there is heterogeneous groups, that is, clusters, and an entire cluster chosen as a sample. Sample size. Sample may be recognized as large sample or small sample. The sample size N is greater than 30. It is known as large sample. The sampling distribution is of normal distributions. If the sample size N is less than 30, it is known as small sample. The sampling distributions are T, F, and chi-square distribution. End of the topic of sampling theory. You can refer to these mentioned books for this topic. Fundamentals of Statistics by S. E. Gupta. Pharmaceutical Statistics, Practical and Clinical Applications by Sanford Bolton. Please take a note on important study questions. Thank you.